My name is Tianna DuPont. I'm a tree fruit extension specialist in Washington State with Washington State University. I'm really pleased to talk to you a little bit today about a project that we recently completed looking at soil health indicators, particularly for Central Washington orchards. You all are probably familiar with the concept of Liebig's barrel. This is a defining concept in plant nutrition where we consider our yield and yield potential to be limited by the lowest limiting factor, in this case primarily macro and micronutrients. But over the past 20 years, our idea about soil function has really evolved, and we're now thinking about the biological and physical properties of soil as well as the chemical properties. So if we continue this concept of a barrel as an illustration, we might have additional limiting factors, for example, root health and water availability, in addition to macro and micronutrients. When we think about soil health, we're generally thinking about how soil health relates to soil function. For example, sustaining plant growth, minimizing erosion, regulating water flow, buffering and filtering potentially toxic materials. When we think about that function of plant productivity, there's a number of things we want soil to do for us. For example, maintain healthy roots, have microbial available nitrogen, have sufficient water entry and movement, sufficient water holding capacity, and competition and protection from pathogens. But it's a little tricky how we measure this. There's so many different potential measurements of soil health indicators, and particularly growers in Washington were interested in which of these many indicators are related to yield and packouts. And so we started a project looking specifically at this. We wanted to identify what set of factors were useful to track in Central Washington orchards, particularly which are related to yield and packout. We want to go from a test like this at left, which has many different factors, perhaps too many to really wrap our brains around, to a fewer number of factors and ones that we know are related to our management goals. The way we went about this was looking at match sites. So we asked growers to tell us about blocks where they had a block that they're just not quite sure what's going on, why it's not meeting their yield goals. And then we found another highly productive block, according to those growers, on the same rootstock, same variety, same general soil type, training system, and age. Of the 100 sites that we looked at, about 60 were well enough matched to be included in all of the analysis. These sites were located throughout the Central Washington growing region where most of our tree fruit is produced. In each site, we took 50 to 100 cores, mixed those up, took our subsample, and sent it off to the Cornell Soil Health Lab for a number of biological, physical, and chemical properties. And then in the field, we also measured water infiltration and compaction, and in my lab looked at nematodes, microarthropods, and root lesion nematodes. Also tried to account for irrigation and crop load. We wanted to make sure that we weren't just seeing water limitation, but rather we're focusing on these soil health related factors. So let's talk about those functions related to root health. One of the measurements that we did is a root health bioassay. This is growing apple seedlings in pasteurized, so heat-treated soil, and comparing to the field soil in that site. We look at the relative height and weight of those seedlings to decide how much pathogen pressure we might have, good guys to bad guys in those soils. In high pathogen pressure sites, you're going to see larger seedlings in the pasteurized heat-treated soil compared to the field soil, and in low pathogen pressure sites, those seedlings will be about the same height. We also did a bean bioassay. This is developed by the Cornell Soil Health Lab, where we grow beans in field soil for 4 to 5 weeks and then rate those roots on a 2 to 9 scale. When those roots are pretty healthy, they're going to be quite white and light colored, compared to when they have more pathogen pressure, you're going to see more of these brown lesions on the roots and the hypocotyl. We're going to be looking at a number of these types of figures, so I just want to explain this quickly. On the x-axis, we have what we actually measured, in this case the root health bioassay for beans, and on the y-axis we have a score. This is a percentile score on a 0 to a 100 scale, where 100 is going to be a good score, like your math test in high school, and 20% not such a good grade. In some cases, this is just based on the mean and standard deviation, and in other cases, we have an optimum score based on prior knowledge of that metric. When we look at the bean root health rating, 29% of the fields we looked at had low root health ratings with a 5 to 9 bean root health score. With the root health rating, 30 of the blocks we looked at had seedlings where there was a big difference in the height between the seedlings grown in the pasteurized and the non-pasteurized soil, indicating potential root health pressure in those sites. Why does this matter? Well, let's look at an example here. Figure you see a relatively high yielding site, 60 bins per acre, and a lower yielding site, 40 bins per acre for that grower. They're both Gala blocks, and you can see that in the lower yielding block, those trees just never really filled their space. This is a particularly bad section of this block. And in that lower yielding block, the root health rating was quite a bit lower than it was in the higher yielding block. We also looked at root lesion nematodes. Those have this needle-like stylet they use to pierce into those plant roots. It disrupts the cell tissue. They're not able to take up water and nutrients as well as allowing other fungal pathogens easier entry. You can see this root lesion nematode has that needle-like stylet that it's going to use to pierce into the roots, disrupting that cell tissue and making it more difficult for that plant to take up water and nutrients. Of the sites we looked at, 15 of the 101 sites had high lesion nematode ratings. That's where we have more than 80 lesion nematodes per 500 cc's, which is what we would consider to be potentially constricting for young orchard sites. Here's an example of where that was probably important. Figure in the lower left-hand side, you can see those trees are smaller. We have some gaps, that patchy distribution that's common with nematode pressure. And in that site, that root lesion nematode rating was lower than in the higher yielding matching block. In this case, it was 129 lesion nematodes per 500 cc's, above that 80 lesion nematode threshold. And you can see you also saw a difference in that bioassay where the seedlings from the low site were smaller than in the pasteurized soil. But not all nematodes are bad. We have fungal feeding nematodes, bacterial feeding nematodes, as well as predators and omnivores. Looking at all of those different nematodes in the soil give us an idea of the structure and function of the rest of the soil food web. For example, we have an index called the structure index that looks at the numbers of omnivores and predators in the soil compared to the rest of the nematodes in the soil. And this gives us an idea of how well structured our soil food web is. And we had a variation with some sites high and some sites low across the sites we looked at. An important function, of course, is nutrient availability. This is in terms of plant productivity. And not surprisingly, in our sites, we generally had high macro and micronutrient levels, with the exception of some high pH as well as a few excessively high phosphorus levels. And this isn't surprising only because in these particular sites, we'd ask growers for blocks where they're not quite sure what the limiting factor might be. And so we're not surprised they already had their macro and micronutrients generally where they should be. The biology in the soil, they're performing many different functions for us, but two primary areas are nutrient cycling and pest suppression. One way to look at the biology in the soil are the microarthropods. These are the mites and springtails that live in the very surface part of the soil. Are important for a number of soil functions. For example, orbatid mites eat fungi and algae and dead plant material, and that That decomposition then releases nitrogen available to the plants. Mesostigmatid mites are predators that are going to help with pest suppression in the soil. More than 10 sites had more than 20,000 microarthropods per square meter, which is a lot of nitrogen mineralizing potential. Several other factors of microbial available nitrogen, for example, potentially mineralizable nitrogen. These measurements of available nitrogen are really important because they're showing the amount of nitrogen that's slowly available over time, and if we ignore that nitrogen, we could be over-fertilizing. For example, potentially mineralizable nitrogen of 21 micrograms per week, which was our average, would supply 50 pounds of nitrogen over a 20-week season. We also looked at respiration. Respiration is the amount of CO2 that's released as the microbes breathe or respire. It's generally a measurement of the amount of microbial activity in our soils. In most of our sites, that number was relatively low, with only 10 sites with high respiration levels. An important function in our central Washington orchards is related to water. We are in an area where we have to be irrigating, and so the water holding and drainage capacity of those soils is really critical. We looked at a number of different indicators, including organic matter, bulk density, compaction, texture, and available water capacity. Remember, available water capacity is the amount of water that a soil can hold between when it's at field capacity, when it's been fully irrigated or rained and had a chance to drain but not dry, subtracting out the water that's so tightly held to those soil particles that it's not accessible to plants. That number is generally going to be higher in a loam or a clay loam soil compared to in a sandy soil site. In 16% of the sites that we measured, we had either moderate or severe water limitation based on that available water capacity measurement. Here's an example of why that could matter. This was a pretty extreme site. This site had 94% sand and also had a number of other potential limiting factors, but you can see that those trees just really were having a hard time, and the bins per acre were pretty unacceptable. Of where that might matter. This is a large block of Granny Smith, and in one half of that block we had 64 bins per acre. In the other half of that block we had around 34 bins per acre. You can see that in the lower yielding section those trees are just not quite as vigorous, not filling as much of their space. And in the lower yielding section of the block, the percent sand was higher and the available water capacity was lower than in the higher yielding section of the block. Soils is their soil structure. One way we measure that is looking at compaction. So above 300 psi, generally penetrating the soil. And that means that they're not going to be accessing water and nutrients as readily, and that can lead to pooling and lack of drainage. We measured 26 of the 101 sites, and 5 of the matched comparisons had areas with compaction that was likely limiting plant growth and productivity. A number of other measurements of soil structure, including bulk density and water stable aggregates. Compaction might have been important. This is blocks of Ultima Gala. These blocks were right across the orchard row from each other, planted the same time on the same rootstock. And you can see that we did have a compaction layer in the lower yielding block. Generally, the compaction layers that we found were at 12 to 18 inches rather than as deep as we found here or in the surface levels of the soil. In some sites, that compaction also led to water pooling, which was like in root health measurements in those sites.
Soil organic matter is related to many functions of soil. Includes the living, dead, and very dead material in the soil. The active pool of organic matter is the freshest organic material, plant and animal residues that have just begun to decompose. This dynamic part of soil organic matter tends to be highly related to nutrient supply, improved soil structure, water infiltration, and stimulation of microbial activity. It is a measure of the small portion of the organic matter that can serve as an easily available food source for soil microbes, thus helping fuel and maintain a healthy soil food web. One way to measure active carbon is permanganate oxidizable carbon. This is a field demo adapting a laboratory technique. First, you add 2 ml of permanganate to your vials. Then add 18 ml of DI water to each tube, filling to the 20 milliliter line. Then add 2.5 grams of air-dried sieved soil to each vial. Shake for 2 minutes at about 2 strokes per second. Let stand for 10 minutes, allowing the soil to settle. Add half a ml of the liquid from the upper part of the suspension to a second tube of 50 ml of distilled water. The permanganate oxidizes the active carbon and loses some of its color. The more active carbon found in the soil, the more the purple color declines. This color change is measured with a spectrometer or a colorimeter. Here you can see that the orchard soil with more active carbon is a lighter pink color. Another measure of active carbon is the particulate organic matter in the soil. Here we have washed the soil through a series of screens and removed particulate organic matter. You can see that the soils with more organic matter additions have more particulate organic matter. Organic matter additions such as compost, organic nutrients, grass clippings, and wood chips are important to build the active carbon pool of our organic matter, as well as stable organic matter over time.
Measurements was active carbon. Active carbon had a wide range, with 57% of the sites having less than 2% organic matter. Of these indicators is useful to track in central Washington orchards, and particularly which were related to yield and pack out. Well, to get at that, we needed to get at yield, so we asked growers for their previous 3 years of yield data and got average measurements. We also took in-field measurements for a subset of sites and determined packed boxes per acre, subtracting out defects due to things like sunburn and bitter pit. A linear mix effects model to determine the association between yield and each of the selected soil health factors, which showed strong trends in exploratory analysis. Available water capacity with a threshold of 0.15 and percent sand with a threshold of 70% had significant relationships with yield. With a threshold of 80 and the bean root health rating with a threshold of 4 had consistent but not significant models with similar results for packout. A model which combines the factors for each of these variables that are most important into 3 factors: water availability, root health, and nutrient availability. We now have a model that we can test for orchard systems. In this model, each factor affects yield only after it goes past a designated threshold. The factors are cumulative. For example, if water availability and root health are both low, the impact on yield is additive. The model is nonlinear because the factors can be related. We now have a draft minimum data set of soil health indicators for central Washington orchards. We think, based on this recent work, that we want to include indicators of water availability and root health, as well as nutrient availability based on grower management goals. Due to those large amounts of nitrogen available slowly over time in a lot of the soils we looked at, we think we should include an organic nitrogen availability indicator. With 25% of our sites showing subsurface compaction, compaction is going to be another important thing to test. Although organic matter wasn't related to the grower management goal of yield in our analysis, it is related to many other functions of soil, including microbial activity, water and nutrient cycling, as well as filtering of potentially toxic materials, and thus should probably be included as a factor related to environmental health. With that, I want to thank you all and thank the growers who very graciously allowed us to sample in their sites and gave us input on the project, the Washington State Tree Fruit Research Commission for their funding, as well as my colleagues Lee Kalsitz and Clark Kogan who worked with me on this project. Have a lovely day, you guys.